Good evening. Thank you for joining us tonight to hear Dr. Goffredi's talk. I'm Lisa Collins, and I'm the president of Scas. I joined the board in 2013, and I've enjoyed serving on the board ever since. I'm sure I've met many of you at the annual meetings, either in talks or poster sessions or during the plenary sessions. I've thoroughly enjoyed planning annual meetings, chairing those symposia, judging student talks and posters, and working with the board. We're really excited to have you all join us again for tonight's event, and we hope to continue spotlighting our board members with these talks. Our hope is to increase your connection to SCAS and show you the value in continuing to support SCAS and our mission to support the science and scientists working in Southern California. For our guests, if you enjoy this talk, we would encourage you to consider joining SCAS as a member. We do offer a student rate. We hope to see you at future SCAS spotlights and at this spring's annual meeting, which will be held in a similar virtual format. Now I'd like to introduce our moderator, Ms. Jade Morris. Ms. Morris is a graduate of the SCAS Research Training Program, which she did for 2 years. She traveled with the research training program to present her work at the National American Junior Academy of Sciences meeting in Austin, Texas, and also in Washington, D.C. In 2019, she presented this research at the International Workshop on Symbiotic Copepoda in Russia. She's currently a student in the College of Creative Studies, focusing on biology at the University of California, Santa Barbara. And now I'll turn it over to you, Ms. Morris.
Thank you, Dr. Collins, and hello, everyone. Welcome to the first BioSpot SCAS lecture series. My name is Jade Morris, as she said, and SCAS truly has really helped me kickstart my career in the science world. I'm honored to be the moderator for tonight's event, and to those of you in the audience, there will be a Q&A text box at the bottom of your screen, and we will be pinning your questions throughout the talk, and I will try to ask as many of them at the end as I can. So without further ado, I would like to introduce the speaker for tonight, Dr. Shana Goffredi. Dr. Goffredi got her bachelor's in biology and marine science at the University of San Diego, and then got her PhD at UC Santa Barbara, working on the physiological functioning of the hydrothermal vent tube worm Riftia pachytilla. To be honest, Dr. Goffredi has truly been a major influence on me and has been an inspiration to many young scientists. She will be talking about her captivating work in the deep sea. And the title of her talk for tonight is The Relationships Between Deep Sea Animals and Bacteria. So please take it away, Dr. Goffredi.
Thank you, Jade. And thanks everyone for being here. It's a really great pleasure to be able to tell you about some of the research that we've been doing at Occidental College. And as Jade mentioned, I titled this Extreme Alliances because the animals I'll be telling you about today are truly extreme in their willingness to form relationships with bacteria. And they're also extreme in the place that they thrive on the planet, which is, of course, the deep ocean. So let's talk about the deep ocean a little bit. It was for a long time considered to be azoic or devoid of life. And of course, we know that that's not true. And it was about 170 years ago that we realized that there was amazing animal life in the deep sea. But it really wasn't until about 50 years ago, I guess when I was about 6 years old, that people discovered that there was animal life thriving and it was abundant and it was fascinating and vast. So the deep ocean represents over 96% of the habitable space on Earth, and its healthy functioning is crucial to the well-being of the rest of the planet. And we spend most of our time thinking about this tiny little sliver of inhabitable space, and the rest of it is obscured, of course, by the surface of the ocean. So I'm going to take you below that surface today. Many of the most recently discovered animal families have been found in the deep ocean. So this is not even just species, single species. These are entire families of animals. And I have one behind me here. This is like a giant Yeti crab hug, you see. This animal is one of the animals I'll be telling you about today, and it's an entire new crab family. It was not known even just 10 years ago. We have better maps of the surface of Mars than we do of our own ocean floor. So there are some estimates that we have only mapped about 15% of our planet and the ocean floor. And of course, it's time consuming and expensive, but there's a real concerted effort now with drones to actually map the rest of it. And so we really do look forward to that, having those roadmaps really. So it's really exciting to be a deep sea biologist, and the deep sea is really a frontier of science and discovery. And there's so much more to understand about this planet. Much of it remains undiscovered. So of course, there is trouble on the horizon, and there's been trouble in the past. And human activities in the deep ocean are accelerating. And that can, of course, negatively impact those ecosystems. And this ranges from climate change to fisheries to pollution. And then, of course, also deep sea mining. So there are precious metals at the bottom of the ocean that are being targeted for exploitation. And this is gold, copper, nickel, manganese. And of course, this is inevitable because of the technology that we have today. A lot of our cell phones and others use these precious metals. But we hope that, at least deep sea scientists hope that the exploitation of those global resources can be done responsibly. And so that brings me to this animal. This is the scaly foot snail. Its scientific name is Chrysomalin squamiferum. And we discovered this animal in 2001 in the Indian Ocean. And this was a giant expedition with a lot of people on it. And this was a very exciting discovery. And it's also a new group of animals. And it became in 2019 the very first animal to be declared endangered by deep sea mining. And that's because some scientists got together and proposed to the International Union for the Conservation of Nature, the IUCN, that this animal might be considered to be endangered if deep sea mining would commence. And the reason why they approved this proposal is because this animal only lives at 3 underwater volcanoes in the Indian Ocean in a space that matches maybe 2 American football fields, that surface area. And so it has a very narrow habitat range. And should deep sea mining occur, it would be a threat to this animal. And 2 of their 3 habitats, so they live only in 3 places in the ocean, 2 of them are currently under mining exploration licenses. So again, it's inevitable, and we hope that they involve scientists in trying to understand the impacts of deep sea mining before it happens. So you might wonder, why should we care about a tiny little snail at the bottom of the ocean? Well, there's many reasons, but I'll just give you 2 of them. One is this animal is unlike any other on Earth. There is no animal on Earth that looks like this animal, acts like this animal. And in fact, the only one that we could find that is even comes close lived 500 million years ago in ancient seas. And this is a limpet called Halkieria. Also, though, this little snail caught the attention of material scientists and the military, and they're currently exploring this animal. It has really amazing armor, and they're thinking about including some aspects of its armor into body armor in the military. So there's a true sort of knowledge reason for understanding and protecting these deep sea habitats, but then there could always be an applied reason to protect them as well. You never know. So my research program really focuses on like the weirdos of the deep sea. And these are many animals that we study in my research team, all with undergraduates, I might add, at Occidental College. And I'm going to share with you 3 deep sea vignettes today. And we study these animals from the perspective of symbiosis. And so these animals have teamed up with bacterial partners in many sort of very intimate ways to have some kind of superpower that allows them to live on this planet in ways that we didn't predict animals could live and let alone thrive. And so I really think about symbiosis as being synonymous with innovation. So these animals are morphologically unique. So they are innovating their body structure and form. So you can see that some of these animals probably look very mysterious to you and look unlike any other animal. And so they've made new tissues to accommodate bacterial symbionts. Sometimes they've enlarged certain tissues to accommodate them. And so they're very good hosts of their bacteria by evolving new tissues. They're also biochemically innovative. So some of these animals have metabolic capabilities that we really never predicted animals should be able to perform. And they're doing this again to care for their bacterial symbionts that in turn, of course, care for them. I should have defined symbiosis, by the way. So symbiosis is really, at least in my lab, we define symbiosis as a beneficial interaction, mutual beneficial interaction between, in this case, a marine invertebrate and usually a single bacterial partner. But you'll see in one of the examples I'm going to show you multiple bacterial partners. Excuse me. So because of this morphological and biochemical innovation and this superpower of teaming up with bacteria, these animals are also innovative ecologically. So they are living in habitats on Earth that it was not thought that animals could survive and be so successful. And so they're taking advantage and exploiting habitats that really aren't available to most animals. And then, of course, as a consequence of that, symbiosis is a powerful force for adding organismal diversity to the planet. So think about how diverse these animals are in all of their novelties, and they are then adding more diversity to the planet. So it's a really, really powerful force, symbiosis. So just to give you some sense of the time frame of these relationships. So I'll tell you that these animals have been in these relationships for about 60 million years to 200 million years, depending on which of those animals you're looking at. And so I wanted to give you a sense of how long that has been. So talk about commitment. So this is 60 million to 200 million year commitments. And this is the amount of time that these animals have been interacting with these bacteria. So just to give you a perspective, this is our planet, and we'll go in order, in reverse time here. So this is about 60 million years ago, 100 million years ago, just to put this time frame into perspective. And I'll stop it here. So this is about 190, 200 million years ago, when all of the continents on this planet were a single supercontinent surrounded by an enormous ocean. That's how long these animals have been interacting with these bacteria and sort of honing these relationships. So it's a pretty long time. All right, so let's talk about explorations of life in the deep sea. So how do we even get down there? Of course, we have to use submersibles, and these animals are 1 to 2 miles deep in the ocean. And the Atlantis is one of the ships that we use quite often. It's home sweet home for many of us for 4 to 6 weeks on end. And this is the Alvin submersible, the research submersible. And this is a submersible that I've had the great fortune to use many times in my career. And the very first time I got in Alvin was in 1994 as a graduate student at UCSB, Jade. So we're having similar life histories here. And then I've been using the Alvin ever since. And so it's been a really amazing submersible to use. We also use another platform run by the Monterey Bay Aquarium Research Institute, and this is a robot. So this time you're seeing scientists in a control room looking at what the robot sees, and you see that this is a giant underwater volcano, and where the scaly snail would be living. And this is also a really great platform. We can have the robot sample for us. You see all the navigation equipment there. So you get the sense. It's kind of like being in a movie theater, but you're watching a robot on the seafloor. So and this is a remote vehicle instead of the human occupied vehicles. All right, so the first little animal vignette is I'm going to take you off the coast of Costa Rica. So what you see here is the bottom of the seafloor. So this is with all of the water removed, and the color is showing you the depth. So red is 0 meters depth, so at the surface, and then purple is 4,000 meters depth, so it's pretty deep. And you are staring into your screen at the capital of Costa Rica, San Jose. And you see, we were very interested in this tiny little feature called Jaco scar. And it is a little scar, and this had been discovered long before we came along. And it's a little feature of slumping of the hillside underwater. And what you should also be drawn to is how heterogeneous the seafloor is. So there's all these seamounts down there. And this is called a convergent margin, where one of the plates of the Earth is actually subducting under the other one. And so it's shaving off the tops of those seamounts as it subsides underneath. And so it is creating a really unusual habitat where that occurs. And those are the habitats that we're very interested in. And these are called methane seeps. So in these areas, there's methane and sulfide emanating out from the seafloor, and we get really unusual animal life living there, many of which are in symbiosis with bacteria. So these are sort of the common ones. There's some golden mussels that you see here, some longer tube worms, some snails, maybe you see some bacterial mats. But we were very interested in a different worm that was very unusual. So some of the people on the ship were worm specialists and noted that these animals looked a little bit strange, and they were also acting a little strange. They were in places in and amongst the known chemosynthetic animals. So chemosynthetic meaning that they're relying on chemical energy for nutrition rather than the sun, energy from the sun. And so what we could tell, and these are all the sort of hazy green that you see in this left-hand image, and those bright white calcium carbonate tubes, those are the animals we were interested in. And so some of our scientists on board thought that they were accumulating in high densities, and they also maybe looked a little bit different than they usually did. So we collected these animals and we discovered their species identity. So the top one is called Lamina tubus, and it's a serpulid tube worm. And the lower one is called Bispira, and it's a sabellid tube worm. And so I'll be telling you just about the top one, even though both of them are quite amazing and both symbiotic with bacteria. So because of time, I'm skipping a few steps here. I'll just tell you that we had a sense that these animals were using bacterial symbionts for nutrition. And we could tell that based on looking at isotopes of their tissues. We had also measured the microbial community. We analyzed the microbial community associated with these animals. And so we understood that there was a single dominant bacterial player sort of hanging around these worms. And so then the next step is to do microscopy. So we knew the identity of these bacteria. We could take the crown tentacles of these animals, and we could do fluorescence microscopy. And we could light up the bacteria and make them fluoresce green. So what you see here on the left is the blue nuclei of the host cells, the worm cells, and then the green filamentous bacteria that were the symbionts. And so you can really get a good sense of the ultrastructural arrangement between bacteria and animal. So they basically have these fuzzy heads covered in bacteria. And this is I direct some of the students who are on the call to a recent publication that we have on this amazing animal. So the other thing you can do is look at them via transmission electron microscopy. So you can send electrons through the tissue and you get this amazing two-dimensional reconstruction at a very fine scale of what the animal and the bacteria look like and how they're interacting with each other. And so you see here that I've labeled all the bacteria, and we can tell that those are the bacterial symbionts because they have these laminated membranes inside of them. And these are bacteria that eat methane. And so that was what was so exciting, is that these worms are actually harnessing methane via a bacterial intermediate. And so these bacteria are known to eat methane. And we had to then figure out, well, you've got this bacteria hanging around with this worm. Are they involved in the nutrition of the worm? So if you're going to invoke a bacterial symbiosis with a marine invertebrate and you want to say it's nutritional, there are other things you have to do besides just identifying bacteria that are associated with animal bodies, of which there are many, and doing some microscopy. And so this next step, I will say, takes a village. So many people have to be present to launch one of these expeditions. It's all the way from the ship's crew, the pilots, of course, the captain, all the scientists, and the chief scientists that write the grants that get the money, the principal investigators, but then all the administrators on shore. So it really does take a village to even answer this one tiny little question about whether or not these bacteria are providing nutrition for these worms. So it's a huge endeavor by a lot of people. So we did some on shipboard experiments. And what we wanted to do, if you're trying to look for whether or not an animal is using a chemical energy source rather than eating on its own, what you do is you actually label the chemical source and you feed it to them and you see whether or not they kind of incorporate it into their bodies. And so we trapped some worms in some jars here, and we fed them some methane that we could track through their tissues. And I won't go into the details here, but we were trying to figure out whether the worms actually could rely on methane. No animal on Earth can use methane on its own. So if this animal is relying on methane for that carbon, for its nutritional source, it's doing it because it has bacterial symbionts that can do it. And then, of course, taking advantage of a totally novel niche and nutrition source. And so what you see here, all I want you to notice here is whether or not the experimental animals, which are in dark purple, shifted to the right from the control animals that were also in jars but not fed the same methane, and the native animals that were collected from the seafloor. And so you can see, we just want to see a shift from a carbon-13C signature in their tissues from around, minus 55 here, we want to see a shift to the right. And if the shift to the right means that they were eating that special methane that we gave to them. And when I say eating, I mean via their symbionts. So that was confirmed that they were incorporating this carbon from the methane into their tissues. And so we really do think that these animals are making this happen via their bacterial symbionts, but we didn't know how. And so then we went back to this more traditional microscopy and we started looking more carefully. And what we saw was that these animals are actually engulfing, it's like they're eating the bacteria at their the skin or the epidermis of their crown tentacles. They're not using their mouth. They're like absorbing them like a blob of slime. And so what you can see here is these arrows are showing you the bacteria sort of being engulfed in these pits. And then we can actually, if we look deeper into the worm tissue, we see that those bacteria are getting incorporated. And so they're farming their bacteria from their heads. And there's only a single type of bacteria sitting on their head. So this really is probably a relationship between the two. The bacteria probably get nutrients from the worm as well, but we don't know what that reciprocal nature of the symbiosis is. But this was an exciting discovery because we had not ever seen serpulids, worms within this worm family. They're not known to host symbionts. They're also not known to be chemosynthetic or rely on these seeps or vents like this, these underwater volcanoes. And it also has ancient implications because for the longest time, some people who study these paleo-oceanographic features have realized that there are these fossil serpulid seeps. So that's the name of these worms. And they've been very mysterious. So they pile up and sometimes they're still underwater, like off of Los Angeles here in Santa Monica Bay, you see these pictures, but sometimes they've been pushed up onto land. So you can actually visit these sites on land. And what these scientists were noticing was that there's tons of these serpulid worms interspersed with traditional sort of chemosynthetic animals like clams that we have always known have bacterial symbionts and they rely on them. And so nobody could really figure out what is the reason for these massive amounts of these tube worms, these serpulid worms in and amongst the clams. And now we think we solved the mystery because they actually are also chemosynthetic themselves. And you can even, we've been to this site with the ROV and we've grabbed some of those tubes and you can tell by some subtle features of the tubes, these old calcium carbonate tubes, that this actually is the same genus, Laminatubus, as the one that we discovered off of Costa Rica. So has some ancient implications for why these worms have been found in this fashion in these paleo seeps. So pretty exciting. Okay, next animal vignette is the Yeti crab, the famous Yeti crab. So this is the very first ever video that was ever captured of the Yeti crab. And this is Kiwa hirsuta. We discovered this animal off of Easter Island. So boy, about as far as you can get from any continent, way off in the Pacific. And so this animal was at 2,207 meters depth. So you see the readout. This is from the Alvin submersible and dive 4090. And you can see this is a magnificent crab. And this animal, of course, only the two scientists and the pilot and the submersible realized that this crab was new, and they've brought it up to the ship, and we all realized it was new. And you can see the animal there on the right. So this animal was collected, and it is the holotype. So that means it is the designated the designator of its own species and its own genus and its own family. of crabs called the Kiwaiidae. And there were many more. So this makes it look like there was only one crab down there and we collected it, but there were many others. Since then, other populations of Yeti crabs have been discovered. They're different species. They're all within the same genus. This is the dancing Yeti crab, also from Costa Rica, called Kiwa puravita. And this was discovered by scientists from Scripps down in San Diego. And they've published extensively on this population. So we've been, and that's the animal that's behind me here. We have been over to Costa Rica to study this population quite a bit. And I'll tell you why they're dancing in just a minute. So this Yeti crab is the ultimate bacterial farmer, even more than those worms I just showed you. So you can see that all over this animal's body are bacteria. And the one that's most striking, of course, is the white filamentous bacteria that make this crab look hairy. And that's why we named it the Yeti crab, because it looks like the abominable snow creature. And so that is not, those little hairs are not the crab's hairs. Those are actually filamentous bacteria. You can also see bacteria on their antennae at the top left. You can flip them over and look at their abdomen, and they have a bunch of bacteria in their abdomen. And so we're pretty familiar now with the community of bacteria that lives on the Yeti crab. It is a novel community that just likes to hang on to crustaceans. And I just want to make a plug here for symbiosis and the microbiome. So if you think about the fact that there are maybe 10 million animals on Earth, only described, there's estimated to be many, many millions more, but 10 million animals on Earth, species, all of which host some bacteria. You can see how much biodiversity on the planet is just contributed to by looking at the relationships between bacteria and animals. And these are beneficial relationships, not harmful relationships. So think about the biodiversity on the animal bodies in addition to the animals themselves. Okay, so why are they doing this? Why are they so furry? It turns out that they actually selectively graze their own bacteria. And this is an amazing video taken by Andrew Thurber and his team when they had first discovered Kiwa puravita off of Costa Rica. And you can see, so think about like your grandfather's old comb that he pulls out of his pocket. They have that kind of structure on their mouth part. So they basically can just comb their bodies. And that comb is unusual for crabs. They normally don't have this comb-like structure. So you can see that Kiwa is really just a agricultural farmer growing its own bacterial fur coat in order to eat it. And I have tried to offer them other kinds of food when they've been in captivity on board ship, and they won't take any other food. So they only will feed on their bacterial fur coat. And they do this in rapid fashion. So we were out at sea in October of 2018 off of Costa Rica, and I kept these Yeti crabs in a tank on board, and we were doing some experiments with them. And after 5 days, this is the same exact Yeti crab, you can see that they completely denuded their body of all their bacteria. So they consume nearly all of their bacterial symbionts every 5 days, I estimate. And so now we're very interested in how these Yeti crabs sort of keep their bacterial community lush and growing like a perfect farmer. And we now know that they do this by dancing. So they basically dance to grow their food. And the reason why they're dancing is that they're helping their bacterial symbionts get all of the nutrients that they need. So their bacteria need oxygen and sulfide and maybe some methane. And so they move their arms around and wave them around in order to provide all of those dissolved gases to their bacterial symbionts. And a really amazing study by a group at JAMSTEC in Japan showed that the movement of the Yeti crabs and these hairy crab types really does stimulate the metabolism of the bacterial symbionts. So we know that they're dancing in order to stimulate their bacteria. So it's a pretty interesting relationship. So my group is still studying Yeti crabs. We're very interested. I have some undergraduates working on them now. So we're interested in a few things. We're interested in the ontogenetic variation in bacterial symbionts, which means what happens throughout the lifetime of the crab, so during development. So you can see up at the top left here, there's an egg, and that's brooded by the mother. And then there's a hatchling crab, and then there's like the teenager crab, and then there's the adult. And all the while, these crabs have to molt. And so we're really interested in what happens to the microbial community when they molt, because they essentially have to shed their exoskeleton, and you think they're with the symbionts because they're on the outside of the exoskeleton. But right away, the young crabs get their symbionts back. And so we don't know how that happens. So is there a little community of bacteria that sort of is left behind to seed the new microbial community, or is it something else? They do hang around in we call them parties of Yetis. So that maybe they get them from other adults like termites, who knows? We're also interested in the fresh molts and the microbes that stick around. And this was for the first time we saw a newly molted Yeti crab. So you see this beautiful, freshly molted Yeti crab on the right. And this was because we were teaming up with some of the microbiologists who were studying free-living microbes in rocks, and they were doing these incubation experiments with their rocks for many days. And we just threw in a few tiny little Yeti crabs to see what would happen. And one of them actually molted. So we got to retain the molt, which is what you see on the left there, the discarded molt. And then you see the new juvenile. And so you can see it's a little bit bigger. Its sort of aspect ratios are a little bit different. So it's growing and changing a little bit. So we're really excited to have that opportunity to look at newly, freshly molted Yeti crabs and see what happens to their bacterial community. So those are some of the questions we've been still pursuing. Okay, and so the last vignette here is about Monterey Canyon. So this is off of the California coast, and this is at 2,900 meters depth. And this was, for all the young scientists out there, this was complete luck, total serendipity. The submersible literally almost tripped over this dead whale on the seafloor. We were looking for something completely different, and it has changed the careers of many of us, this one discovery. And so you see we're zoomed in here on this amazing red-headed animal, just kind of flowing in the ocean currents there. So you can see that this is a dead whale, and you can see the bones and you can see the vertebrae really clearly. You might also see some crabs, some worms. This is like a bonanza of food, like a giant deep sea buffet. But what drew our attention, and this is one of those times when we were using the robot, the remote vehicle. And so there was a whole bunch of us in the control room just screaming at the screen, jumping up and down, like, what is that? Because nobody had ever seen an animal like this before. And it turns out it is still a pretty mysterious creature. So of course we grabbed one of the rib bones of this decomposing whale. It's called a whale fall. This happens, of course, because whales die during migration. It's pretty hard for especially the young whales to get from Mexico to Alaska and back. And, Monterey is a whale migration corridor. So you would expect some whales to succumb and then sink to the seafloor. And this has got to be the weirdest underwater habitat ever. I never thought I'd be studying dead whales on the bottom of the ocean, but it happens to have an animal that I've just fallen in love with, and it is symbiotic with bacteria. So what you see is that the red and the white, so we cut this rib bone in half, and what you see is the red and the white that sticks up into the water column is really the tip of the iceberg. The worm goes way down into the bone with this crazy tissue. So talk about morphological innovation. So this is one of those animals. It turns out that this animal, well, at first we could not even place it into a phylum. So that's a very high level taxonomic group that even the 12 scientists that were in the control room cannot even place it into a phylum. And so we finally involved some experts. We did some molecular analysis and some morphological analysis, and we realized that they were a group of polychaetes, a very derived group of polychaetes, worms. And we named them Ossidax, which means bone devouring in Latin. And all the ones you can see with the naked eye are female. And so you can see that this animal is very strange, has a really strange green root tissue that's unlike any other animal on Earth. And inside that green root tissue is ovaries, and then these long blood vessels that go down into the roots and sort of permeate the bone. And as I mentioned, they're all females, but of course there must be males somewhere. So the males turn out to be hanging around on her trunk. And so they live here, and she has hundreds of microscopic males that hang onto her with these very sinister looking hooks. And then they clamor around to fertilize the eggs as she lets them go out into the water column. So this is the male. The males are 500 microns in size. So they're essentially a little larvae that never really develops, but develops mature sperm so it can fertilize the eggs. And the female is 4 centimeters in length. So this is a huge size difference among these two animals. And this happens a lot in the deep sea. So if you don't have much of a chance of finding your mate, it's best to keep them nearby in miniaturized form, which happens in some deep sea animals. So I'll just tell you a couple things in the last few slides here about the amazing Occidental and our research trajectory. We've been studying this animal for nearly 20 years, and there are still many major mysteries about this animal. But I'll just tell you what some of the things we do know. So we made this discovery in 2002, as I mentioned, totally by accident. We described a couple of species of Occidental, Frank Presi and Ruby plumus. And then we realized that these animals didn't have a mouth or a gut or any way to feed themselves. And so, of course, my symbiosis brain, of course, it had to be a bacterial symbiont. So we looked in the weirdest tissue we could think of first. And of course, that was the green root tissue. And we found the symbionts in this green root tissue. Again, this is a collaboration by many, many people in different disciplines. And we realized, not like the very first worms I showed you, that they had bacteria hanging off of their heads and they kind of consume them. And not like the Yeti crab that also has bacteria on the outside of their body. This animal, these worms are letting bacteria inside their cells. So the most intimate that an animal can get with bacteria is to let them, it's very, there's a lot of negotiating and committing that has to happen, but they let them all the way inside of their cells. And that's why you see them in this sort of round structure here. Also same fluorescent technique that I explained before. Same transmission electron microscopy technique that I also showed you. So you can see an animal cell all the way around the outside where the arrowhead is, and then you see bacteria all within that cell. So these worms have not only created this novel tissue, but they are supporting bacteria inside of their cells, and those bacteria are providing nutrition. And then you zoom in even further and you can see a little bacteria inside of a vacuole. So not only are they inside cells, but they're inside of vacuoles. And so there's a lot of coordination, like molecular dialogue, that has to happen between this worm and these bacteria that are inside of these vacuoles. And you can also try to look at root tips to see how much of the biomass of the animal's green tissue is made up of bacterial symbionts. So in this fluorescence image, you see that the bacterial symbionts are actually in red, and the green is the animal. And so you can see quite a bit of the biomass of the root tips is devoted to bacterial biomass. And so we realized we had this conundrum. So we weren't the first people to ever study whales on the bottom of the ocean. In fact, people studying whale falls, they had been doing that for a long time, these ecologists. But something really weird was happening with this whale fall in Monterey Canyon. So we had read about whale falls that could sustain life for like 50, 100 years. But you see in just 5 years, this whale fall has essentially been almost completely degraded. And so what we realized, many people had been studying some of these whales then that had Occidental on them, that Occidental essentially accelerates the degradation of a whale fall by mining the bone. That's how they're getting their nutrients, via their bacteria. And so they're actually doing an ecosystem service in the deep sea by making that sort of recalcitrant, hard to break down carbon available for the rest of the deep sea. And so if Occidental is present on these whale falls, they essentially degrade within 5 to 10 years. So that was definitely new in the whale fall ecology world. And so I'll just, I'll give you one last slide of 2 of my most favorite discoveries about Occidental, and not by our group. So this was we always had this conundrum. So we thought that Occidental was a 60 million year old worm that only ate mammal bones. But marine mammals are only 30 million years old. So we had this 30 million year old age gap that we could not account for. And then finally, some paleo scientists were looking at prehistoric marine reptile bones, and they were doing cryotomography scanning, and they realized that they could identify Occidental burrows. So this is like that ovary and that green tissue and the start of the trunk, that they found Occidental burrows in these prehistoric marine reptiles. So that actually meant that Occidental could be as old as we assumed them to be, because before whales, they were probably feeding on marine reptiles. And since then, of course, we've put turtles and crocodiles and turkey in the bottom of the ocean, bones, of course, and Ocedex will happily live on those bones as well. So we've empirically shown what they discovered. And then finally, many scientists have discovered at least 25 total Ocedex species worldwide. So you might think this is like a one-off thing that we discovered in Monterey Canyon. It's bizarre and weird, but it's the only one you'll ever find. It turns out that there are many, many Ocedex species worldwide. And one of my favorites is called Ocedex java, because it's the most inflated of all of the Ocedex. And so it resembles Jabba the Hutt. And this one has been very elusive because even though they do live on bone, they're always sort of obscured in the mud. So you see that they're really, really hard to find. So we've been looking all over the world for them, and we've still only found them in Monterey Canyon. But they've been discovered in the Antarctic and the Indian Ocean. So they really are worldwide. Okay, so I will end there by saying that this, again, takes so many people to be involved, but it's such a fun process. There's funding by the National Science Foundation and all of the people that help, including my amazing research team at Oxy, and then all of the scientists at Caltech, at Scripps, Temple, all the people that made all of these cruises possible. Shout out to everybody on board. And then I'm happy to take questions. I'll leave it here with the octopus high five. We were not reaching out to touch this octopus. This octopus came to us in this very rare moment of marine invertebrate meets giant metal object in the seafloor. Thank you for watching.
Awesome. Thank you so much. This is such an amazing talk. I feel like I learned so much. So some of the questions that I'm seeing, the first one here is, what attracts you to an area that makes you explore there looking for interesting biological things?
Yeah, that's a great question. Usually we're looking for heterogeneity on the seafloor. So we like this hardscape. So this is a perfect image maybe to answer that question. So all of this rock in the background is called carbonate, and that is made by the activities of microbes. And so, of course, if you are mapping the seafloor, you can see that there is actually hardscape instead of mud, and we use sonar to do that. And so we would probably go to areas that have interesting sonar features in order to look, but also those slumps can be very interesting because you imagine that a lot of the chemicals that are buried in the seafloor become revealed when you get like a slumping feature. So that scar, like I showed you, was I'm sure that the Germans who very first discovered that site were really making a beeline for that feature on the seafloor.
Awesome. Okay. This next question is from Rebecca, and it is, do you have to take into account pressure when incubating the worms?
Yeah, that's also a great question. So I did my PhD on the giant tube worm Riftia pachyptila, and we did physiological experiments on them. And so we actually repressurized them on board the ship to make sure that the physiological experiments and measurements we were doing were absolutely accurate for what the worm was experiencing on the seafloor. It turns out that these worms, we did not see that they suffered too much coming up to the surface, but we only did our experiments for 5 days. So I do think that you are limited in how much you can push these animals, because like you said, or maybe you're imagining, extreme is kind of in the eye of the beholder, right? So when we bring them up to the surface, this is now extreme for them. We think the deep sea is extreme. They think up here is extreme. And so they are exquisitely adapted to those pressures based on their enzyme functioning and their lipid membranes. And so some of them do suffer more than others by being in atmospheric pressure. In this case, we did not, we repressurized the bottles a little bit, but just with gas, we didn't actually take them back down to the pressure we collected them from. So you do have to take the data with a grain of salt, especially if you're going to do like RNA transcripts and metabolic functioning, for sure. It's a good question.
Okay, so the next question is from Deborah Leopo. So it says, I love your enthusiasm for deep sea organisms and microbes. Do you know how the microbial symbionts are being acquired on the Yeti crab or the lamnitubious worms, horizontally or vertically?
Oh yeah, good question. Yes. Let me see. So we don't know about the lamina tubus because we haven't found eggs. So I'll just give a few definitions that she gave. Vertical means that the new hatchling invertebrates get their symbionts from their parents, one or the other, and horizontal means they get them from the environment. And there's both in the deep sea. So you might think it's really risky to hatch and have to find your symbionts from the environment. Occidental does that. We know absolutely for sure that Occidental gets their symbionts from the environment. With the Yeti crab, it's possible that it's pseudo-maternal or pseudo-vertical because the female crab will brood the eggs, and just by the virtue of her holding onto those eggs near her abdomen, it's possible that her symbionts can make their way onto the eggs, which we do see that they're present on the eggs, and that then they're present when the little organism hatches. So I would call that pseudo-vertical because there's still some access to the environment. But with the lamina tubus, we have no idea. Still have no idea. Yeah.
Awesome. Okay, so Maggie here asks, thank you for the great talk, Shana. Are there challenges to studying live deep sea inverts at the surface, e.g. exposure to lower pressure, potential air exposure? And would you expect there to be any differences in behavioral or symbiotic processes?
Mm-hmm.
Do you feel they feel stress at the surface?
Yeah, there are definitely challenges. So I'd say the biggest thing that we have to contend with is keeping them at the right temperature. So temperature, not matching their temperature is way worse than not matching their pressure, it seems. These habitats, so the hydrothermal vents are different. They're a little bit elevated, but not hugely elevated where the animals are living. But at these habitats, like you see here and where lamina tubus and the Yeti crabs are, it's cold in the deep sea. And so you do have to, we keep them in insulated containers all the way up to the surface. And then we never expose them to air. And they have to be kept cold the entire time and then back into whatever pressure vessel or experiment that we're going to put them in. So for sure, that can be a stressor on them physiologically. Yeah, it's a good question. Yeah.
Oh, the question. Okay, there we go. So Dean asks, you discussed many symbionts today. Do you think that these organisms will be more threatened by climate change or better able to adapt to changes due to the bacterium's faster timescale for evolution?
Oh, that's an interesting question. I think that any relationship that's already been built over the last 200 million years is going to be under threat, and one that maybe not be as nimble as you think. So certainly the bacteria have faster turnover rates, but the animal, by taking some nutrients from them, they don't have as fast of turnover rates as a free living microbe would have. So there is a little bit of a slowdown in the symbiont's replication. Otherwise, they would just overgrow the animal. And same with your own microbes. There is some study, so we're not sure about how these animals are going to fare in the future. Certainly, ocean acidification is going to harm any animal with a calcium carbonate shell or an endodermal skeleton like sea urchins and sea stars. But there have been studies on coral reefs where you obviously are aware of bleaching. There are some corals that are forming relationships, so they'll spit out their old symbionts and reacquire new symbionts of a different type that can fare better in warmer temperatures. So that is a much more sort of nimble and flexible relationship between cnidarians and their algal symbionts. It's not clear whether these animals, they're not very, I would say it's called promiscuous in their uptake of symbionts. They are very faithful to the particular partner, and it's not like they can just swap symbionts when times get tough. So we're not really sure yet. It's a great thing to think about, though.
Okay, so then the next question here comes from Gordon Hendler. It says, fantastic, Shana. Do all Yeti crabs dance to grow food?
It's a very good question. So there's 4 species of Yeti crabs that live at methane seeps, where the chemicals are just sort of gently kind of effusing from the surface. There is one species, the very first one we discovered, that was living at hydrothermal vents, the explosive underwater volcanoes off of Easter Island. That one does not dance. And I think it's because the water is already so dynamic and billowing around down there that maybe it's not necessary for them to dance. So we discovered the very first yeti species and did not remark at all on it being a dancer. And then when they discovered the Costa Rican population, it was all over the news that it was this dancing yeti crab. And so we're like, we never saw ours dancing. Really, we didn't. And so we really think that it's the seep ones must dance and the hydrothermal vent ones don't have to. Yeah.
All right. Julie Passarelli asks, she says, awesome talk, Shana. Hi, Julie. How long are you typically out at sea, and how long does it take you to get to the various sampling locations?
Yeah, that's a good question. So the Easter Island and the Indian Ocean cruises were very long. So they were 5 to 6 weeks. So you really do, and you don't see any land, hardly at all. And so you spend quite a lot of time. We often spend maybe 5 to 7 days off of our coast here in California. So we have another cruise coming up in May to study these seep habitats off of our coast. So it kind of just depends on how remote the location is. And if you mean how long is the descent to the bottom of the seafloor, and with a submersible, for example, it's about 2 to 3 hours. Then you spend a few hours on the seafloor, and then you have another 2 to 3 hour ascent. So it kind of depends on the depth, but my longest time in Alvin was 9 hours.
Wow. All right. So Jose asks, can you discuss how Osedax disperse? Is there another habitat besides whale fall sites?
Yeah, that's a really good question. So we can make all kinds of artificial sites on the seafloor that we know that Osedax will get on. So the adults don't move. They are embedded in the bone and will never move again. But they do spawn egg and sperm that fertilize in the water. And so that forms a larvae. And those larvae are slightly negatively buoyant. So we think that they go down to the seafloor and they kind of swim around a little bit, but they only probably can last 3 to 4 weeks surviving on yolk. So they're not a free feeding larvae. So they have to find another habitat, suitable habitat within 3 to 4 weeks probably. And but we have implanted whales on the seafloor that have washed ashore, and within a month we get a thriving Osedax colony. So we do think it's kind of just a numbers game, kind of like the dandelions of the sea. The females will spawn a whole bunch of eggs and fertilized eggs, and then it's just some will find a suitable habitat and some won't. And it's just, as you know, if she can put out as many as possible, then some will be successful. So that is how they move around. But I will say that the larval lives of these deep sea invertebrates remains very mysterious. It's incredibly hard to study the life cycles of these animals. And there was an entire National Science Foundation program called the LARV program that tried to do just that. And some people made some great strides in our understanding, but it's really very mysterious.
All right, so we have about 3 minutes left, so probably only about 1 or 2 more questions. This here is from Carl. Hi, Carl. Have any planktonic larvae for deep sea invertebrates ever been found higher in the water column? Would it be possible for the bacterial symbionts to travel with larvae to a new site or vent?
Oh, yes. Certainly larvae do. So there is, diel migrations of animals up and down the water column, and you can find them, even some of the vent shrimp, their larvae end up in the surface ocean. And they can travel with their bacterial symbionts for sure, but they mean to be living on the seafloor in these very suitable habitats. So they need to find a chemosynthetic habitat. It's not always clear how they know that they found the right habitat. So it's kind of related to the last question too. Do they sense some chemicals? Do they actually, I forgot to mention this, Osedax, we don't even know when they acquire their symbiont as the larvae. Like, are they making their way towards a whale fall and they detect the right bacterial symbiont? So they're like, this must be the place because I sense my bacterial symbiont, and then they settle? Or do they settle first and then just risk that maybe, hopefully, their symbiont will be one of the many, literally, needle in a haystack of the bacterial situation, will they find their correct symbiont. So I think they can definitely move around quite a lot on ocean currents, entrained in the axes of the spreading centers, especially for hydrothermal vent animals. But they really do need very specific habitats, but they can travel many, many kilometers, maybe hundreds of kilometers. Yeah.
Wow. Okay. So last question here. Pauline asks, basically, do you know what determines the sex ratio skew in Osedax?
Well, I definitely think it is because it's in the deep sea. So anglerfish do this as well. And there's some echiurine spoonworms. So miniaturizing the males is actually a strategy in the deep sea. And you can look this up. So in Osedax does have probably the largest, it's called sexual dimorphism of any animal, meaning the female is much, much, much larger than the male. But I think it's important to have as many males as possible and as many as she can support in order to fertilize all those eggs. So we also have amazing video of the continuous spawning of eggs. So she has an oviduct that comes up past her body and out past her head, and she will continuously put eggs into the water column, and they're all previously fertilized. So she needs quite a few males there to facilitate the extreme numbers. I think well, I don't want to say, but it's hundreds and hundreds of eggs a minute, something like that. Yeah. So, yeah.
Awesome. Thank you so much. My apologies if we weren't able to get to any of your guys' questions. We really appreciate all of your guys' participation in our very first event. Thank you again so much, Shana. That was an amazing presentation. And thank you to Scas for putting on this awesome event. And then Lisa, do you have any closing comments?
We will let everybody know when we are doing this again, which will be a couple months from now. But a huge thank you to Shana for being willing to be our first speaker and for the fantastic talk. And Jade, an amazing job moderating. So thank you to you as well. And we hope we see everybody back again soon in another SCAS event. So take care and be well.
Nice. Thanks everyone for joining.